So I've mentioned myelin once or twice. Myelination is the process of wrapping an axon with myelin. Myelin is a type of lipid. That high lipid content gives it kind of a white appearance and also helps to insulate the axon. Myelin around the axon tends to be several layers of membrane of the glial cells. Because it's a membrane, it's a lipid, it has that glossy white appearance. The glial cells that do myelination in the central nervous system are the oligodendrocytes, or in the peripheral nervous system, they're referred to as Schwann cells or neurolemmacytes. In the peripheral nervous system, those Schwann cells encircle the axon and wrap it up in layers, forming that myelin sheath. The cytoplasm and the nucleus kind of get pushed over to one side, and it forms a little bump called a neurolemma, which is why sometimes these cells are referred to as neurolemmacytes. A neurolemmacyte can myelinate only about 1 millimeter of axon, so you're going to need quite a few of them for most axons. There are little gaps between the myelinated areas, and these are called the nodes of Ranvier. Here you can see a single axon, little single 1 millimeter portion of an axon, and then we're going to take our Schwann cell, because this is in the peripheral nervous system, and we're going to wrap it around again and again and again, and that's going to electrically insulate and also slightly help protect the axon. The cytoplasm and the nucleus have been kind of pushed to the side and are forming this little bump over here called the neurolemma. Here's another picture of a neuron. You can see the axon here has been wrapped up with the Schwann cells or the neurolemmacytes, and in between them are those gaps that we call the nodes of Ranvier. Those nodes of Ranvier are going to become really important. In the central nervous system, the oligodendrocytes can partially myelinate multiple axons. They send out little foot processes that will wrap up a little millimeter section of an axon. And one oligodendrocyte can myelinate several axons, but just little sections of them. So you can see they all kind of participate in and help to myelinate axons completely. We also have the nodes of Ranvier inside of the central nervous system, just as we do in the peripheral nervous system. In the peripheral nervous system, we do have the occasional axon that is unmyelinated. These tend to be because several axons will kind of be engulfed by one Schwann cell. What the myelin sheathing helps to do in both the central nervous system and the peripheral nervous system is to effectively speed up the signaling that we find in both of those areas. If the speed of signaling is messed up, then that's going to cause a lot of issues with both receiving and sending signals, so both the sensory and motor functions can be detrimentally affected. There's a couple disorders in the central nervous system. We have a demyelinating disorder called multiple sclerosis. It is an autoimmune disorder where oligodendrocytes are attacked by immune cells, and as they're killed off, they're no longer myelinating parts of the axons. This is an inflammatory event, and as these happen repeatedly, it's going to cause scarring and permanent loss of function. Guillain-Barré syndrome happens more in the peripheral nervous system. You get muscle weakness beginning in the distal limbs, and it advances to more the proximal muscles, and these patients tend to make a full recovery. After traumatic injuries, the peripheral nervous system can regenerate. Regeneration is possible if the neuron cell body is intact and if enough of the neurolemma remains. Regeneration is more likely to succeed if the amount of damage is minimal and the distance between the site of damage and the structure it's innervating is shorter. To regenerate inside of the peripheral nervous system, we have our axon that's damaged, and then proximal to that area, the axon is actually going to seal off and swell up a bit. Distal to the cut, the axon sheath is going to degenerate, but the neurolemma is going to survive. The neurolemma and the endoneurium will form a regeneration tube, which, along with growth factor released by the neurolemmacytes, is going to go ahead and help guide the growth of new axon collateral filaments that will kind of fill up the area and then merge together and make a single large axon again that will go ahead and reinnervate the original tissue that it was already innervating. The central nervous system, axons do not regenerate. The oligodendrocytes secrete a growth-inhibiting molecule, and a large number of axons are crowding in the CNS, making repair more difficult. And then the astrocytes and connective tissue will grow within the damaged area and scar the area, preventing the growth of new axon filaments through it. This scarring happens relatively quickly inside of the central nervous system, and this is what prevents areas of the brain or the spinal cord from reattaching. There are drugs that, if administered quickly after a, say, spinal cord injury, can really help recovery, and that they prevent the scarring from forming, allowing any kind of axonal growth to continue.